welcome everybody and welcome Sham Sundar Palchina.
Thank you.
Sham Sundar did his PhD from CDRI. He did it from my laboratory, which he finished in November 2017. And right away, he moved to University of California in San Diego, where he is continuing his postdoctoral research. Sham has been quite productive during his PhD. He has 18 papers, co-authored 18 papers, out of which 2 papers were directly related to his thesis. You may remember the one, particularly Shubhashish may remember, and others in my lab would remember, the one which dealt with globular adiponectin, its function in bone as well as in muscle, and its mechanism of action that involved mitochondrial biogenesis and mitochondrial respiration. And the other paper was on a wakefulness drug, modafinil, which came from the screening of FDA-approved drug library, in which case he showed that this wakefulness drug, which is actually quite widely used even in children who are suffering from
ADHD.
ADHD. Attention deficit syndrome, and in that case it works on adenosine receptor, leading to activation of NF-kappa B and consequent bone loss. And then he moved to, as I told you, after his PhD, he moved to UCSD, and here we will listen from him his work in his new laboratory, and the title is The Role of Protein Kinase G2 in Bone Homeostasis and in Adaptation to Mechanical Loading. Interestingly, in this regard, I think there could be an overlap of what Shubhashish did related to protein kinase A and cyclic AMP signaling in bone in relation to various phosphodiesterase inhibitors, and what Konica is continuing. I think they, particularly these 2 people, in addition to myself, of course, would be interested in hearing his recent research. Sham, please continue your presentation.
Thank you, sir, for the introduction. Good morning, everyone. As the title showing here, I'm going to present my present work, which is related to the protein kinase G2 and its role in bone metabolism. Here I am showing a diagram of the PKG2 signaling, or where usually our group primarily focuses on this protein kinase G. The kinase enzyme, which is activated by elevated level of cyclic GMP. There are 2 isoforms of this protein kinase G. One is PKG1 and another is PKG2. We explore the role of PKG1 in cardiac pathology in our lab, and the role of PKG2 in bone biology is also explored. So my work primarily focused on the protein kinase G2 and its role in bone metabolism and pathology. In this diagram, I have summarized the role played by the protein kinase G in osteoblast and osteoblast lineage cells like osteocyte. So, for example, when external stimulus like mechanical stimulus, estrogen, and thyroid hormone activate nitric oxide synthesis, this is the endothelial nitric oxide synthase, eNOS, which in turn activate the soluble guanyl cyclase, which converts CAMP to CGMP. When the CGMP level increases in the cytosol, that activates the protein kinase Gs, PKG1 and PKG2. The PKG1 and PKG2 are usually expressed in different tissue, like PKG1 is important for the cardiac health, and the PKG2 is in the bone and fat or mesenchymal stem cell originated cells. Activated protein kinase Gs may phosphorylate multiple downstream signals and signaling molecules, which in turn regulate physiological roles inside the osteoblast, like commitment or differentiation, as well as their proliferation and anti-apoptic signaling. So before going further in our present research, I want to summarize few of our previous articles, which actually made the base of this present research. There, I'm showing here summary of four of our previous articles. The first one is published in Diabetes in 2018, where we have shown that sinasigwart, which is an activator of soluble guanine cyclase, activates PKGs. Sinasigwart treatment to the type 1 diabetes mice restored the osteoblast function and bone quality by suppressing the oxidative stress. In another paper published in Endocrinology 2018, shows that the transgenic bone-specific expression of protein kinase G2 leads to higher bone mass compared to wild-type mice, and those transgenic mice are protected from diabetic-induced bone loss. Another paper in 2017 published in JBMR, our group has shown that another NO donor, here the NO donor nitrosylcobinamide actually, when it enter into the body, it releases the NO, which in turn activate the soluble guanylate cyclase. So NO donor nitrosylcobinamide treatment can protect mice from the postmenopausal osteoporosis. And the last paper listed here is the oldest one that has dealt with the mechanotransduction. There, our group has shown that the protein kinase G2, through the activation of AKT and beta-catenin signaling, modulate the mechanotransduction in fluid shear stressed osteoblast. This fluid shear stress is used to mimic the mechanotransduction in vitro condition. This paper was published in 2009 in JBC. So as you can see that in all those papers are showing the same thing and trying to say that the PKG2 is critical for osteoblast biology. So in our present research, we generated a osteoblast specific protein kinase G2 knockout mouse. To generate this knockout mouse, the exon 3 of the protein kinase G2 or PKG2 gene with the 200 BP flanking intronic sequence were cloned between two LOXP sites. And then after making the transgenic floxed animal, we mated each other in the littermates, and then we created the homozygous floxed animal. When the floxed animal is created, we created another Cre-expressing gene, the Cre recombinase, and the Cre gene was put under the Col1A1 promoter, which confirms that the Cre gene will only be expressing in the osteoblast lineage cells. So as a result, when this floxed mice and the Col1A1 promoter controlled Cre gene is expressing, or the Cre gene is present, then those are the knockout mouse, and when only the flox mouse act as a wild type mouse. And we created this mouse in C57BL6 background. To confirm that the knockout is successful and it is specific to the bone, the expression of the protein kinase G2 at mRNA level as well as the protein level, we tested in tibia, brain, and kidney. And as you here you can see, let me start the laser pointer. As you can see in the tibia, in the knockout mouse, the protein kinase G2 expression is almost zero. In the brain, kidney, and intestine, the mRNA level is intact. Here in the B, it is the protein level of the PKG2. It is almost gone. And here in the wild type, it is expressing properly. In other tissues, also the PKG2 expressing properly. Here, this is the immunohistochemical image where the protein kinase G2 positive cells are stained here. While talking about the protein kinase G2 knockout, we also tested the expression of the other isoform, that is the PKG1 in tibia. Here you can see in this PKG2 knockout mice, the PKG1 expression is intact. After confirming that our mouse line is osteoblast specific knockout, and it is our next target was that to elucidate the bone phenotype of this protein kinase G2 knockout mice. So we took 8 weeks old litter mates and we analyzed the bone microarchitecture using the micro-CT scans and compare the protein kinase G2 mice bones and with the wild type litter mates. We selected 8 weeks old litter mates and assessed the trabecular and the cortical bone quality of the tibia. Here, the 3D image represent the tibia metaphysis and the trabecular. Here in the image, you can see the trabecular bone in male knockout mice is significantly deteriorated as indicated by the reduced trabecular volume, or so BVTV. The trabecular number is down. Here the trabecular thickness is low here in the knockout, as well as the bone mineral density is also low. Surprisingly, in the female mice, the knockout and wild types are same. There is no deterioration in the bone quality. The similar pattern we also seen in the cortical bone. As you can see here, the male knockouts showing less cortical area, less cortical thickness, and lesser tissue mineral density. Then we assessed the reduced bone formation parameters in this male and female mice, and we found that, as the image here showing, the knockout male has very fine difference between the two calcine levels. And when we assessed the histomorphometrical parameters like mineralized surface by bone surface, mineral apposition rate, and bone formation rate, only the male knockout is significantly lower compared to the wild type here in the MAR and BFR, but the female wild type and knockout are same. After his hematoxylin and eosin counter stain, we also counted the osteoblast number in the cortical bone. We found that the male knockouts has less number of osteoblast in the cortical bone. The bone formation marker serum PNP is also decreased in the PKG2 knockout male mice, but in female, the knockouts are resistant to any change. I mean, their bone and the bone formation marker all are same in the knockout and wild type. To explore the underlying mechanism, we assessed the gene expression in bone transcriptome. We found cell cycle related gene, osteoblast related genes, and some of the Wnt signaling related genes are downregulated in male PKG2 knockout mice compared to their wild type litter mice, while the female wild type and knockout are indifferent. We further check the beta catenin protein level in the bone lysate, and you can see that the knockout mice, the male knockout mice, has less amount of beta catenin protein in their bone. So from here, we are thinking that the beta catenin, as in our previous paper, we have also shown that the beta catenin signaling is responsible for this PKG2 mediated bone homeostasis. We are thinking that when the PKG2 knockout um, osteoblast or bone, the beta catenin signaling is impaired. So here it is the primary osteoblast where we did this ALP stain. You can see that the male knockouts are lower, uh, low stained. Here their mineralization capacity is also shunted, but in female there is no difference. Then we treated this male and female wild type and knockout mice with CGMP. CGMP usually activate the signaling pathway through the protein kinase G. This CGMP we use, it is 8-CPT CGMP, which is cell permeable and enters into the cell and then activate the signaling through activation of protein kinase. Here you can see that in the male, when CGMP is added, the wild type, the cell proliferation increases, indicated by the BRDO positive cells. In the female, in the wild type cell, addition of CGMP increases the cell proliferation. While interestingly, when in the knockout, whether it is male or female, CGMP signaling is impaired. So after adding CGMP, irrespective of the sex, the proliferation rate decreases, which make us, which help to this indicate that knocking out the protein kinase G2 in the female cells does inhibit the CGMP mediated signaling, but female cells have other compensatory mechanism which helps to overcome the CGMP signaling deficiency. The next part of the project was the role of protein kinase G2 in bone mechanotransduction. This is, I think, more interesting part of this project, and this is a little bit different from which I did in my PhD also. Here, first let me explain the mechanotransduction. The ability of the bone to sense and convert external mechanical stimuli into a biochemical response, which ultimately alters the bone phenotype and its function. Mechanotransduction helps animals to adjust with altered load-bearing activity, whether it is exercise or weight gain. Like you know that with increased weight, the bone mineral density usually increases. Or even walking, if your exercise pattern changes, your bone adjusted with the increased load, so you gain more bones. Bone mechanotransduction is also critical to maintain bone health during the long-term exposure to microgravity, which is a critical part of research in the space science. So here I have tried to explain the mechanotransduction model in mice, how I did it. For the mechanotransduction studies, we have devised a sequential loading machine following a published protocol. We used a commercial actuator which could be programmed using a computer software to produce defined amount of force in a defined interval level. The figure A here, this one, shows how I put the mouse tibia or the mouse leg specifically, because it is a live mouse and the tibia is actually not touching either of the cups. It is the feet or the knee is touching the cup. I put the mouse tibia in the custom made cups, and the lower cups, this one is fixed, while the top or upper cup is attached to the actuator. This is the actuator rod here. So In the left picture shows the whole setup. Like here, the mouse is under anesthesia. Here it is supplying 2% isoflurane. The left tibia or leg was loaded, and the right tibia was not loaded and used as a controlled. The diagram B shows the protocol used for loading. Here you can see that 1 Newton base load is maintained throughout the experiment, and then within 1 second, 8 Newton or 7 Newton force was generated on the tibia. So total 220 pulse of load every day was given to the mice, and we loaded the mice like from Monday to Friday first week, and in the second week again Monday to Friday, and in the third week on Monday the animal was euthanized. And both of the tibia, like the left and right control and the loaded tibia, were isolated and micro-CT was done. So the next question was, and one important thing here, we injected calcine and alizarin on day 8 and day 12 to check the bone growth. The one interesting part of this thing is that the analysis of the tibia is different from the normal tibial analysis. We have to find out that which area or which part of the tibia is actually responsive to the load. As tibia's shape and size is pretty much not uniform, so each part is sense the strain or load in a different way. That's why to decide which part of the tibia need to be analyzed, what we did, we analyzed 3 loaded and unloaded tibia from each male and female wild type mice. Here, the lower line is showing the control or not loaded tibia, and we analyze the cortical thickness or cross-sectional thickness of the whole tibia. And we plotted the loaded bone behind with a higher. And we saw that in male as well as in female, from this 150 to 550, this part of the tibia is most sensitive to the load, and this part, the highest amount of bone growth has been seen. So we chose in our all experiment, we decided to use or decided to analyze this part of the loaded bone to find out how much loading is, how much bone growth is happening. So the next thing is strain measurement, because it is not the load but the strain which decide, or in a simple language, in how much the bone is bending. Strain is a measurement of the bending of the bone. So the strain decides the load response and osteogenesis. As we all know that the load bearing capacity of the female bone is less than the male bone, that means the load required for the female bone to experience equal strain as the male bone should be lower. So we did a strain gauge experiment. Here in this slide, here it is the example of how the strain gauge is attached to the tibia, and then these wires were connected to a computer through an adapter. And when this tibia was compressed with the actuator, it shows similar kind of pattern. It is here we loaded the tibia with increasing amount of force. You can see with 4 Newton, it was showing around 500 microstrain. With 5 Newton, it goes like 650, then 6 Newton to up to 10 Newton. So we loaded 3 male wild type, 3 female wild type, and 3 male knockout mice to compare their load strain ratio. And what we found that at the 8 Newton, force in male mice, wild type or knockout, here you can see they are almost superimposing on each other, 1400 to 1451. So around 1400 microstrain was generated after 8 Newton of force, while the female mice, 7 Newton force can produce around 1400 micro strain of strain. So we choose 7 Newton force for the female mice and 8 Newton force for the male mice, which will generate equal amount of strain, that is 1400 microstrain unit. Here the 3D images shows that where the tibia was actually gained bone. Due to the irregular shape of the tibia, the strain felt by different surface of the tibia differs significantly. So the bone gain for all loading induced bone gain experiment, we calculated the difference of delta of the cortical parameter between the loaded and controlled tibia. So here this is the delta of loaded and control bone. So you can see it is approximately, on average, it is 0.1 millimeter square of it is the cortical area, and it is in the female. Here it is cortical thickness in the male, and it is in the cortical thickness in the female. As you can see here, the cortical area and cortical thickness is lower in the knockout mouse compared to the wild type mouse. But in the female, wild type and knockout mouse, both mouse, the net bone gain is almost equal, insignificant. Then we did the histomorphometrical analysis, and here you can see that only the male knockout mice is not stained properly, or only the male bones show poor calcium leveling, while the female wild type and female knockout both has green and red both of the lines. To find out what might be the underlying mechanism, we loaded the male and female mice for three, and after the loading, we waited 3 hours and then did the qPCR to find out which genes are altered after the induced by the loading, and we found that lot of beta-catenin is definitely increased after the loading, but in the male knockout it is indifferent, while in the female you can see the beta-catenin is increased after the loading in both wild type and knockout. So that means the beta-catenin female mice definitely have a compensatory mechanism. And so from these studies and previous studies, what we summarized here, like the protein kinase G2 loss and gain of function in osteoblast produces striking skeletal phenotypes, and it in turn established the protein kinase G2 as a key Wnt beta-catenin modulator, and it is also involved in the mechanosensing in the male mice, but not in the female. As you know, there are multiple drugs are approved by the FDA, like the riociguat and tadalafil, which usually increases the cGMP level in a different mechanism, might represent novel treatment option for the osteoporosis in male, but probably also postmenopausal females, as we have shown previously in the postmenopausal. While the estrogen is not present in the system, the females may be prone to bone loss when the knockouts if PKG2 is knocked out, and possibly the estrogen is the bypass mechanism in these knockout animals in female. So as you see, that sexual dimorphism we found here, which is unusual. So we further identified a novel crosstalk between the protein kinase G2 and the androgen receptor. And we are presently working in that, which possibly explain the sexual dimorphism in this protein kinase G2 knockout mice. So this is what I wanted to show. And this is the recent paper we published in JBMR, showing all this data I have described earlier. And this is our research team in UCSD Department of Medicine. I directly work with Professor Renata Pils, and these are the collaborator in UCSD Bioengineering Division, Professor Robert Shaw and his team. That's all. Thank you.
Okay, great, Sham. A very interesting work. I'm sure there will be plenty of questions. I myself has plenty of questions, but let us see. Let others ask the questions first. Please go ahead asking questions, clarification, seeking clarification from Sham.
Hi. So obviously really nice work, particularly this PKG2 knockout mice model osteoblast specific could be really helpful if I have this have access to this knockout mice model during my PhD studies. Those sildenafil, vardenafil effect.
The thing is that, just to discourage you, I can understand that before publishing this paper, Mone Zaidi's lab has asked those mice, and my PI, I don't know why, before publishing anything, before characterization, she has given, and they are possibly testing those.
Yeah, that's possible because Mone Zaidi's lab also published a paper with vardenafil effect on
yes,
if
they
they are
have
using,
a CGMP signaling and bone paper. Yeah.
Then probably they are going to publish. But obviously, it's a very nice model and could be really helpful.
Yeah.
So the question that I have is, what could be the possible mechanism behind this sexual dimorphism? Like wind signaling, they are equally present. So is like estrogen mediated wind signaling activation play a major role in female mice to bypass this deficiency?
Yes, actually, estrogen also activate the beta catenin and wind signaling. So when estrogen, and as you know from your experience, estrogen is most powerful hormone in everything. So when estrogen is present, definitely PKG2 mediated signaling is not as significantly strong as PTH or estrogen. So when estrogen is present in the system, the PKG2 knockout doesn't make any problem. But the thing is that in postmenopausal women, when the estrogen is no more in the system, the modulation of this signaling could be beneficial. And one of our NO donor, that is nitrosylcobinamide, has been tested before, and it shows promising effect in the postmenopausal animals, like the overectomized animals. And the second thing is that, as I said in the last line, that we also have found that the protein kinase G2, that paper is just going to be communicated this week, that protein kinase G2 protein has interaction with the androgen receptor. So for the androgen receptor activity, protein kinase G2 is required. So when the protein kinase G2 is not present, the androgen signaling is hampered.
Okay.
Yeah.
Yeah, really nice work. Congratulations on your paper, and thanks for citing our paper also in your publication. I see that
your paper is come across the way, so it have to be published. Yeah.
Any other question? Okay. Sham, your previous research from your lab, I think it was in 2018, where from the title, I haven't read the paper, but the title is very interesting, is that transgenic PKG2 animals have higher bone mass as well as protected from diabetic-induced bone loss. Now,
Yes.
that is very interesting to know that how come with the higher bone loss or higher bone mass,
Yeah.
you are able to get protection against diabetes-induced bone loss. Is this mechanism very much like that of osteocalcin, carboxylated osteocalcin? Is it the mechanism that has been described
The
there?
two things is important here. That protein kinase G2 actually increase the proliferation of the osteoblast. That is my personal experience. I have seen that this transgenic or primary osteoblast expand very fast and compared to the normal osteoblast. And the second thing is they reduce the oxidative stress in the osteoblast. So when the type 2 diabetes mice is type 1 diabetes mice, the oxidative stress in osteoblast and any other tissues also, the oxidative stress is very high. This protein kinase G2 overexpression actually suppress that oxidative stress, and so the cell apoptosis also decreases, cell proliferation increases.
Is it a type 1 model in
Yeah,
which
type 1
perspective?
model. Type 1
Type
model,
1
both of
model
them.
was tested.
Yeah.
Okay.
Yeah.
Since you are getting so much of a proliferation of the
Yeah.
knockout PKG2, you said in so.
No, the transgenic is proliferation is high. The knockout usually proliferates slower.
Okay, so the transgenic is high. That means it is a PKG2 overexpression system.
Yes, yes. Those are primary osteoblast from the transgenic, and the transgenic cell. I mean, the transgene is under the control of COL1A1 promoter.
Okay, so if that is the case, then
Yeah.
and your experience working with these cells is that you find them vigorously proliferating.
Yeah.
Why don't you simply check if those cells have a higher levels of IGF-1 and EGR1? If IGF-1 production is high by osteoblasts produce IGF-1.
Yeah.
And if
Yeah.
IGF-1 production is high, that would lead to a auto-feedback proliferation signaling in osteoblast via EGR1. And these IGF-1, if it is produced in really large amount, it
Yeah.
will mitigate the diabetes condition, insulin-like growth factor 1.
Okay.
So check
Okay.
that. Okay.
Okay. No, I actually plan to do the IGF-1 for the dexamethasone studies, which I have studied very long ago. Here we have also that paper is also in process, and we are seeing that the dexamethasone induced bone loss is also protected in those transgenic mice. And dexamethasone actually suppress the IGF-1 expression. It is known that by other people also published that IGF-1 expression is suppressed by the dexamethasone in osteoblast. So I haven't tasted that. I just ordered the primers. That if the so I can tell you that after that. Yeah, sure. That's a,
And another issue related to your tibia biomechanical testing.
yeah, yeah,
Now, you say that it is 7 Newton for female and 8 Newton for male tibia you
yeah,
apply, and
yeah,
it was given for some 2 weeks in a row.
yeah,
That kind of a regimen you used,
yeah.
and then you studied their response in the knockout animals, male versus female. Now,
Yes,
related.
yes,
Now, what I would like to understand is that in this model of application of strain to tibia, how close it is to the natural system, because our understanding of tibia mechanical loading is that it is basically a torque. It is not bending.
yeah.
There is a bending and tibia is a torque. So what kind of loading I'm trying to visualize that is being applied? Is it mimicking the natural loading scenario?
For mice, it may not be totally, but since we are loading with the tibia is not actually compressed. If I zoom in this picture, you will be able to see this is actually we put the tibia here.
Okay.
So
Panel
it
A,
is,
yeah.
yeah, the femur actually touches the cup here, and here the ankle is touched by the cup, but the not tibia. That is usual what happened to our tibia axillary. So we are not actually compressing the tibia. That is one way to make it more physiological, that not actually we are, it is the wherever the femur and tibia is connecting to each other, we are putting this axial load. But yes, since mice are like four-legged animal, they are not like human, but here whatever the load is there, that is basically how we human actually feel the load more. And
It
there
is
is another,
uniaxial. It is uniaxial, right?
yeah, uniaxial, right.
Yeah.
But
Okay.
because there is another model where they have used 4-point bending, like that is actually
That is done in
totally
tibia, yeah. Yeah,
yeah,
tibia is more like a 4-point bending. Now,
yeah.
why didn't you make these animals run on treadmill?
uh, our plan was not run on treadmill, but we were planning to do something like a tail tail hanging
Oh,
one, that's
yeah,
so
so
unloading.
microgravity kind of a thing, yeah,
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 yeah.
yeah. So,
But
yeah, so that is also another thing people have done, and it is widely used and validated model that you
yeah.
let the animal, but this is very stressful for the animal when you do it.
Yes, I agree. So that's why we started with, actually with, for the male we started with 9 Newton, as other people has shown that that is a good load to show bone growth. Um, so but we found that with 9 Newton there is a injury response, uven bone formation, and that's why we decreased it to the 8 Newton. And at 8 Newton the male bones are growing with like they are forming solid bone, not woven bone, and there is no injury. They are not limping, they are not showing any injury response. That's why we choose this 8 Newton. But we earlier we started from the 9 Newton. And there's another problem with this treadmill thing is uh, in treadmill their both leg will be loaded. And so but here the difference in the it is the cortical bone, it is not basically the trabecular bone. So with the cortical bone, so you see here in the data we have shown the delta of the cortical area or cortical thickness, because if we just put the because each tibia are not their baseline is not equal actually. So their cortical thickness ranges from 150 micrometer to 160 micrometer. So we have to put the delta, like the loaded bone is compared with the unloaded bone of the same animal, and then we get this data. Since
No,
it
I
is a
understand.
cortical,
I
yeah,
understand it, that there are distinct advantages of using this
yeah,
model. But what I'm
yeah,
trying to understand is that since it is not very close to the natural loading
yeah,
pattern, that was the reason I was
yeah.
suggesting of using. Now, if you are why are you so interested in cortical bone? You mean to say that the trabecular bone is not affected? Your data showed that trabecular bone is affected as well.
Yeah, clavicular bone is affected, but here in this uh, here in this loading experiment, trabecular bone is not that, in our model at least. People have shown that their trabecular expression has increased, but in our data only the cortical bone is increased, or cortical bone mass has increased, not the trabecular bone. Trabecular bone between the loaded and unloaded are almost same. There is no much difference.
So quite a few uniqueness in your model then, which needs to be
Yeah,
explained, right? And
yeah,
you have the handful of work then. from this model itself to explain
Yeah.
several things. Okay.
Yeah.
Yeah. Any other question?
Because when we got the reviewers comment, they people are asking mainly like they are saying that even the 19 weeks and 20 our animals were 20 weeks, and they are saying even the 18 weeks animal could have a different phenotype, which is not in our experience. And these models, what I found in the literature, in a different animal, even the C57BL6, the Jackson and Envigo differs. The German, we have C57BL6 from German colony, which is imported from Germany, and also from Japan, and also from Envigo. So the funny thing is the wild type and vehicle treated animal from the German colony cannot be compared with the Envigo or American animal or the Japanese animal, although they are all C57 BL6.
Oh,
So the point I'm saying this, our main aim was to keep it is like the strain specific effect. Like in other strain, you might find that they are also losing trabecular and cortical, not only gaining trabecular bone, they are also gaining cortical bone. But in our system, it is only the cortical bone has been gained.
so let me finally bring this issue to you, is to get a clinical perspective on your finding. Clinical perspective, you have mentioned that in terms of the GMP elevating drugs can be used for senile osteoporosis. I'm not saying about that. I'm saying about what I'm suggesting is that only the male bones are affected by the non-functioning of PKG pathway and not female bone. If you can find from the mutation studies in the literature any kind of echo of your finding in their phenotype.
Yeah. So I didn't get your question. I mean, what is you are asking?
Okay,
like,
my question is that so you are having these sexual dimorphism, and how do you see this kind of, or study this kind of thing in human condition?
Okay, so in human condition, that is what I said, that the protein kinase G2 signaling is important for male, you are saying, but it is also important in the postmenopausal osteoporosis when the estrogen is not present. So sexual dimorphism may be present there, but in the female, the main osteoporosis is postmenopausal osteoporosis, and so it is also usable there in the female.
I'm wondering, I don't know the literature. Can you look into the literature to see if suppose any component of the PKG pathway, any genes in human have mutation, either kind of a activating or inactivating mutation, and if in that family only the male bones are affected or female bones are affected. That kind of study, the mutational study in the human subject, will only be able to tell you its human relevance.
Okay, yeah.
So look literature in these angles, that the PKG pathway, what are the molecules involved in the PKG pathway, and see if there is any known mutation in that PKG in any of these molecules, and if they have studied bone. So a lot of ifs are there, but
I got
if you find any kind of hint from that studies, if available, then you would be able to justify your human relevance very easily.
Okay, yeah.
Right.
that
You
I
have did
can
it
look
in
into.
your last slide towards the end related to the pharmacological
Yeah,
intervention, but your study, what you show here, needs validation at the genetic level in human, whether or not
yeah,
there is even male-female dimorphism in PKG signaling with respect to bone
yeah,
phenotype in human. You have to do some good literature search and see if you get something that will
yeah.
also help you in defending your studies, because
Okay.
anybody would say that, okay, particularly when you say that there is strain specific effect in mice, means people will sometimes say that, okay, if it is even in mice it is strain specific, do you have any human relevance?
Yes, I got it. Yeah.
So try to find a human evidence which can be really which would really strengthen your this beautiful work. Otherwise, people
Yeah.
would say that it is a mice effect,
Yeah. I,
right? That is what I'm saying.
that is true. Yeah. Yeah.
And the way to find, as I told you, is to look for the mutation studies in human.
Yeah. I mean the human studies. Like I
Human
was reading. I
studies,
mean in yeah.
yeah. All the
Okay.
genes, or any mutation that
Sen
leads
or
to
SNP variant.
leads to modification of PKG pathway, you know, not only just
Yeah.
PKG signaling related molecules, but any gene as a downstream of which rise PKG, and that PKG modification
Hmm.
is causing skeletal phenotype, is the kind of search you have to do.
Okay. Yeah, I got. Yeah,
Do that.
I understood.
Any other question? Otherwise, I think it is late at night at Sham's place. We have to tell him good
That's fine.
night.
It is just
And thank you very much, Sham.
9:45. Yeah.
Yeah. Okay.
Welcome.
Good night.
Thank you for this opportunity to present my work.
It was
Thanks a
it
lot.
was very nice work. Congratulations.
Thank
Congratulations.
you.